Welcome to the weekly Neurology Deep Dive. This is an educational analysis focusing on a recent study relevant to your daily clinical practice in neurology. Just a reminder, there are no commercial goals here. This is purely a summary. We strongly advise neurologists to consult the full published paper for complete details. Today, we're looking at a phase 3 trial from the New England Journal of Medicine. It investigated Inebilizumab for generalized myasthenia gravis.
That's correct. The study specifically included individuals with generalized MG who tested positive for either anti-acetylcholine receptor antibodies or the anti-muscle specific kinase antibodies, MuSK.
And our goal today is really to pull out the key findings on efficacy and, of course, safety for this specific patient group using Inebilizumab.
Maybe just a quick backgrounder first. Generalized myasthenia gravis, as you know, it's an autoimmune condition. It causes fluctuating muscle weakness. And this weakness stems from autoantibodies targeting usually acetylcholine receptors. That's about 80% of patients. Or in about 7 to 10%, they target muscle-specific kinase, MuSK.
And B cells are really central here, aren't they? Plasmablasts, plasma cells, they're producing these autoantibodies. We have treatments like glucocorticoids, other immunosuppressants, but, well, some patients need other options.
Exactly. And that's where inebilizumab comes in. It's a monoclonal antibody. Its job is to target and deplete CD19 positive B cells. And importantly, these include the very cells, the plasmablasts and plasma cells, that produce autoantibodies in myasthenia gravis.
Okay, so let's dig into the study design itself. Phase 3. Double-blind, randomized, placebo-controlled. It was conducted across, 127 sites in 20 countries? That's quite extensive.
It is a robust design. They enrolled 238 participants. All had generalized myasthenia gravis, MGFA class 2, 3, or 3 or Phi, so moderate to severe disease. And crucially, they all had confirmed positive tests for either the AChR or MuSK antibodies.
What were the entry criteria in terms of disease severity scores? I recall there were specific MG-ADL and QMG requirements.
Yes, participants needed an MG-ADL score between 6 and 10, with over half being non-ocular symptoms, or an MG-ADL score of 11 or more, and they also required a QMG score of 11 or higher at screening. Just to remind everyone, MG-ADL measures daily living impact, QMG quantifies muscle weakness.
And these patients were already on treatment, weren't they?
That's right, they had to be on a stable dose of glucocorticoids or nonsteroidal immunosuppressants, or sometimes both, when they entered the trial. Reflects clinical reality.
Then came the randomization, 1-to-1, intravenous inebilizumab, 300 milligrams on days 1 and 15, plus an extra dose on day 183 for the AChR positive group.
Correct. The other group received a matching placebo infusion.
And the treatment length differed.
It did. 52 weeks for the AChR antibody-positive participants, but 26 weeks for those with MuSK antibodies.
The protocol also pushed for a glucocorticoid tapering, I believe?
Yes, there was a specified tapering schedule. The target was getting patients who started on higher doses down to 5 milligrams or less per day by week 24.
But what about rescue therapy, if someone got worse?
Rescue therapy was allowed for disease progression. That included intravenous immune globulin IVIG or plasma exchange. But importantly, it excluded increasing glucocorticoid doses.
Okay, let's talk endpoints. The primary one was?
The primary endpoint was the change from baseline in the MG-ADL score at week 26. And this was for the combined population, so both antibody groups together.
And key secondary endpoints.
Those included the change from baseline in the QMG score, also at week 26 for the combined group. Plus, they looked at the changes in both MG-ADL and QMG scores at week 26, specifically within each antibody subgroup, the AChR positive and the MuSK positive patients separately.
All right, let's get to the results. What did they find for the primary endpoint in the combined group?
At week 26 for the combined population, the Inebilizumab group showed an average MG-ADL score reduction of 4.2 points. Compare that to the placebo group, which had a reduction of 2.2 points.
So a difference of about 2 points. Was that significant?
Highly significant. The adjusted difference was minus 1.9 points, with a p-value less than 0.0001. A reduction of that size, around 4 points, often represents a really meaningful improvement in daily activities for patients.
And the QMG score, the measure of muscle weakness.
Similar story there for the combined group at week 26. Inebilizumab group saw a reduction of 4.8 points, Placebo group, 2.3 points. The adjusted difference was managed 2.5 points. Again, highly significant. P less than 0.001 suggests a clinically relevant drop in weakness.
Now, breaking it down by antibody type, let's start with the acetylcholine receptor antibody-positive subgroup, MG-ADL scores.
At week 26, in that AChR positive group, MG-ADL dropped by 4.2 points with inebilizumab versus 2.4 points with placebo.
Yeah.
Statistically significant, P equals 0.002.
And the QMG score in that same AChR group.
Also significant improvement, a reduction of 4.4 points with inebilizumab compared to 2.0 points with placebo. P equals 0.001.
Okay, now for the muscle-specific kinase, the MuSK antibody-positive subgroup, MG-ADL change.
For the MuSK-positive patients at week 26, the MG-ADL reduction was 3.9 points with inebilizumab compared to 1.7 points with placebo. This difference was statistically significant, P equals 0.03.
But what about the QMG score in the MuSK group? I recall that was different.
That's right. For the QMG score in the MuSK group at week 26, while the reduction was 5.2 points with inebilizumab versus 3.0 with placebo, however, that difference, an adjusted difference of number 2.3 points, did not reach statistical significance. The p-value was 0.13.
So, significant MG-ADL improvement, but not QMG improvement in the MuSK group. Any thoughts on that?
Well, it could be a few things. The MuSK subgroup was smaller, so maybe the study lacked power to detect a significant difference in QMG specifically for them. Or perhaps the treatment effect manifests slightly differently in MuSK-MG. It definitely raises questions for further study.
Did they look longer term, say at 52 weeks?
They did, specifically for the larger acetylcholine receptor antibody-positive subgroup. And yes, at week 52, they observed greater sustained improvement in both the MG-ADL and QMG scores with inebilizumab compared to placebo. That suggests a durable effect in that population.
What about clinically meaningful improvement? Did they look at responders?
Yes, they looked at the proportion achieving at least a 3-point improvement in MG-ADL at week 26 without needing rescue therapy. Around 69% in the inebilizumab group hit this mark, compared to about 48% in the placebo group.
That's a notable difference. And the steroid tapering goal.
That was quite successful across both groups, actually. A high percentage, somewhere around 85 to 87% in both the inebilizumab and placebo arms, managed to reduce their glucocorticoid dose to 5 milligrams or less per day by week 26.
Which is great news regarding potential long-term steroid side effects.
Absolutely. And pharmacodynamically, the study confirmed what you'd expect. Inebilizumab led to rapid and sustained depletion of circulating CD20-positive B cells. That fits its mechanism.
Okay, crucial aspect. Safety. What did the profile look like?
Overall rates of any adverse event were fairly similar, around 80% in the inebilizumab group, about 73% in the placebo group.
Any specific adverse events stand out with inebilizumab?
The most common ones reported in 10% or more of the inebilizumab group were headache at 15% and COVID-19 infection at 11%. Remember the trial timing.
What about serious adverse events?
Serious adverse events were actually slightly lower in the inebilizumab group, 8% compared to 13% in the placebo group. Adverse events of special interest occurred in 10% with inebilizumab versus 16% with placebo.
Any major safety signals like anaphylaxis?
No. Importantly, there were no reports of anaphylaxis or serious hypersensitivity reactions in either group.
Were there any deaths during the trial?
There were 3 deaths reported overall, 1 patient in the inebilizumab group and 2 in the placebo group. Various causes were cited in the paper.
So, pulling it all together, what are the main implications for clinical practice based on this trial?
Well, the evidence from this phase 3 study suggests inebilizumab is an effective option for generalized myasthenia gravis. And that seems to hold for patients positive for either AChR or MuSK antibodies, improving daily function and reducing weakness severity.
And the efficacy was shown alongside successful steroid reduction, which is a major plus for patients.
Definitely. Achieving benefit while enabling glucocorticoid tapering is clinically very significant for long-term management.
And the safety profile.
It appears generally manageable, and it seems consistent with what's been seen with inebilizumab in studies for other autoimmune diseases like NMOSD.
But long-term data is still needed, right?
Absolutely. Research is ongoing. There's an open-label extension study underway, which will be key for evaluating the longer-term benefits and, of course, any potential risks associated with sustained treatment in MG.
So this deep dive really highlights the potential of targeting CD19-positive B cells with inebilizumab. It looks like a promising therapeutic strategy for generalized MG.
Indeed. The data from this trial provide valuable information for you as neurologists when managing patients with this often challenging condition in your daily practice.
And just to reiterate, this summary is for educational purposes only. It doesn't replace the need to really dig into the full study yourself. Treatment decisions always have to be tailored to the individual patient circumstances.
So thinking about this data, perhaps you might consider how Inebilizumab could fit into your treatment approach for generalized MG. Are there particular patient profiles where you might consider this therapy earlier or later? It's worth reflecting on. We really encourage you to read the full article, consider all the details, and think about the broader implications for how we manage myasthenia gravis going forward.
Thank you for joining us for this weekly neurology deep dive. We look forward to exploring more impactful research with you next time.